Okay, we'll resume our discussion on protein sorting with this slide, which summarizes a lot of information. The steps are numbered and described here. Proteins being synthesized, being sorted from the endoplasmic reticulum to the Golgi, migrating through the Golgi, and then for one case a secretory vesicle, dumping the protein contents to the exterior, also to and this would be a regulated secretion. This occurs only when a signal happens. You can also have constitutive secretion, which means that as soon as you get to the membrane fuse, proteins can also be sorted to lysosomes. We'll see the process of endocytosis, joining in endosomes where they will eventually go to lysosomes, and the cell will recover some materials and also the retrograde transport from stacks even back to the endoplasmic reticulum, which can occur for one reason if proteins get missorted and they get further along in the process than they should. There is a mechanism to bring them back for another attempt at sorting. Anyway, you should know all 9 of these steps very thoroughly for the tests. Slide, we look at the process of endocytosis. Particles, such as in this case an energy-rich LDL particle, will bind to the LDL receptor. Once bound, the bound cluster receptors will cluster together. Clathrin will start to form on the outside or the inside wall of the plasma membrane. This will encourage an invagination, a pinching in of the membrane, which will eventually fuse in an ATP-driven process. Now the coated vesicle, the clathrin-coated pit, will start to migrate inwards. The clathrin will come off the vesicle, and you'll have a pure vesicle called an early endosome. It will fuse into a late endosome, which has a altered pH. This would be close to physiological pH, 7.4. At pH 5, the receptor no longer has strong affinity for the particle. The receptor, which you can reuse over and over again, will then get sorted to the plasma membrane, released, and it's available to bind more of its LDL particles. The LDL particle itself will migrate into a lysosome where fats, cholesterols, and amino acids will be harvested for energy and for biosynthetic needs. This cycle of receptor movement in and out of the membrane occurs about every 10 minutes. So in this slide, we see how the protein clathrin is going to drive the invagination of the membrane. The orientation is switched now. This is the outside. This is the inside. Here we have receptors which have bound to their ligand, their cargo molecule it's called. It could be a hormone, whatever. That's going to cause an association between the receptor and this clathrin complex. And as the receptors are driven to interact closely because of thermodynamic contacts and adapter proteins on the inside, that necessarily will bring these clathrin proteins in closer contact. And you see, because of the dimensions, that they're wider on the outside than they are on the inside. For them to be close packed, that's going to naturally create a curvature here to the membrane, which will then proceed into the stalk-like structure, which will then get pinched off. And now you have a clathrin-coated pit with the bound receptors bundled here inside the vesicle. Then the vesicle will become uncoated, and this vesicle will travel to the endosome for deposition of its contents. But this is a very clear instance where the shape of the protein is going to drive the function that it's designed to carry out. And here we see the way that the stalk gets pinched off. This is an ATP-driven process by the protein dynamin, and this is very much a boa constrictor-like mechanism for squeezing the plasma membrane such that the continuity is broken. In contrast to endocytosis, there's one other mechanism by which certain cells take up certain species from the environment, and that's called phagocytosis. And in this case, rather than an invagination from the clathrin-coated pit, we grow the membrane around, in this case, a bacteria. This is an immune cell with actin, which will polymerize and form these extensions. It will grow around the bacterium in this case. Eventually, this particle will enter into the membrane, and it will be a double membrane coated species, and then this will go to an organelle referred to as the phagosome. On this slide, we see the organelle of the lysosomes, and this is the principal site of intracellular digestion of biopolymers. You will recover from materials taken up. You'll recover nucleotides, you'll cover amino acids, you'll cover sugars, you'll recover fats, and these can then all be used for energy or biosynthetic building blocks. Now these enzymes, an enzyme that degrades DNA or RNA, or enzymes that degrade proteins, these are of potential danger to the cell, right? You don't want a nuclease getting free, wandering into your nucleus and start chopping up your DNA. So these proteins have a very interesting property. They only function at acidic pHs. So the inside of lysosomes are maintained at a pH of about 5. The cytosol, most of the world around many organisms and in the nucleus have normal pHs around 7.2 to 7.4. And these proteins, for instance, if a lysosome would accidentally rupture and they would spill out into the cytosol, it's true these proteins would get out there, but they would not be active at a normal pH. So this is a safety mechanism for regulating the activity of these otherwise nasty proteins. So the manner in which the pH is lowered inside the lysosome is an ATP-driven process. This is the opposite of what occurred in the inner mitochondrial membrane, in which case you have a protons going against with the concentration gradient, causing the synthesis of ATP from ADP. In this case, we have ATP hydrolyzing to ADP, pumping protons against the electrochemical gradient, with an excess of protons in here being used to reduce the pH of the lysosome. And then on this last side, there are just 3 different routes towards getting material to the lysosome. We have endocytosis, this is the principal one. We talked about phagocytosis, which you'll start to kill bacteria using chemicals, but then it will fuse with late endosomes into a lysosome. And then you have a process called autophagy, which is a process where double membranes are built around organelles, like in this case a mitochondria, which has outlived its utility. It's no longer functioning appropriately. This will become bound and then recycled to the lysosome for other cellular use. Okay, thanks for listening.